Recording now. All right. So good afternoon, or actually good morning, guys. It's 11:30, Friday, April 24th, and this is biology. So what we're going to do today, guys, is we're going to wrap up talking about protein synthesis, and I'm going to show you on our Schoology page what's left in the module. Most of you are where I need you to be, so let me just show you what that is. Okay. So on Schoology. Wrong page. On Schoology, show you updates. So on our updates, I have been posting every day. I post the YouTube link for those of you that can't attend. And like I was just saying, is if you know you're busy or you have other things to do, like just watch the video, know what you missed, and do the work. Okay. But I really do like you being here because I like doing activities with you guys in Zoom groups and all that. So thank you again for those of you that can make it. And like I was saying, it's the same people that keep missing. So I emailed all of them. I have been emailing all of them. And just reach out, okay? And please message me if you have any other questions that I don't answer here today. So there are a few assignments left on the module, and the one that I have scheduled for today is this reading assignment. Only one person has actually done that assignment fully, and that was Christian. Everyone else, again, don't stress on the date, okay? I just wanted to highlight it so that you know it's here, and highlight Christian because, like I said, you can work at your own pace. You don't need to wait for me. So it's scheduled here for today. And then for next Monday, I had scheduled a discussion board, but a lot of you already did it, which is fantastic. Okay. So there's 3 more assignments left. Okay. It's this reading assignment, it's the discussion board, and then it's a quiz I had scheduled for next Wednesday, but you don't have to wait for me. You can do the quiz whenever you want. You just have to message me and I'll turn it on for you. Okay. So any questions about those 3 assignments, which is the reading assignment on Newsella, the discussion board, which a lot of you just started. I got yours today, Jessica. Thank you. And then the quiz. Any questions about those, guys, before I move on?
Yeah, what's the writing prompt for Newsella?
Good. Thank you, Felipe. And actually, to help you with that, I think it'd be more helpful if you saw it from a student page. So, Christian, can I ask you for a huge favor? Christian, can you do me a favor?
Yeah.
Can you turn on? Can you share your screen with us? Felipe has a question like, how do you find the Newsella prompt? And you're the only student that's actually completed it fully. Can you share your screen with all of us? And I'm recording this, so for students that are missing today's class, they can see where to find it, because I think that's been a common issue, Christian, and some people can't find it. So would you mind sharing your screen and showing Felipe how to get to the prompt, pretty please?
All right.
Okay, thank you. I hear you typing, Christian. Are you opening it up? There you go. Thank you. All right, cool. So you see, so did you, hold on, Christian, just pause for a quick second. So Felipe, and this is for everyone, okay? First of all, you have to have opened up Newsella. So Christian, did you go to newsella.com or did you go through Newsella on Schoology?
I went to newsella.com.
Okay, so there's really 2 different ways you can do this, guys. You can go straight to newsella.com like Christian did. And Christian, can you highlight at the top your URL? Just highlight newsella.com. Okay, so you can just go straight to that website, guys. I put it directly on Schoology, but it's also found in your website folder. So however you get there, Felipe, and everyone else that's listening, it doesn't really matter how you get there. But once you log in, Can you go just to your home screen, Christian? Go ahead and get out of the article. I just want him to see, and everyone to see, what this looks like when you first log in. So go to the very beginning. Okay, so when you log in, guys, you see where it says your assignments. Click on that, please, Christian. Okay, you see on your assignments. So it looks like I'm the only person that's using Newsela for Christian. Is that correct, Christian? No other teachers use this, right?
Yeah.
Okay, so however, if, let's say, who's your English teacher, Christian?
Miss Nelson.
Okay, so if Miss Nelson was using that, you would have other articles, but can you highlight where it says teacher E. Trujillo, Christian? Can you just hover over that? There you go. You see how that's my name, Felipe? And you see how it says class honors biology?
Yeah.
So that's how you can tell who's assigning what to you. So for me, the only article I've assigned for this module that we're on, which is protein synthesis, this is the article. And I sent it to you as a PDF, but I told you guys the other day, please don't just use the PDF. Use this assignment. So now can you click on it, Christian, first? Click on gene editing. So when you click on gene editing, you'll notice that I have the instructions here, which are the same ones that we read the other day. So if you didn't watch my YouTube video, please watch it because we read this. Go ahead and click on the article. There you go. And then so go ahead and click on where it says activities, Christian. So once you click on that, it opens up on pause right there. It opens up all these things. But in my instructions, did I say you have to do the quiz?
No.
No, you don't have to do the quiz. And go ahead and click on view assignment, Christian, for those people that aren't listening. Click on view assignment. Highlight the instructions again, Christian, because I have a feeling some people are not paying attention. Click on, or just highlight view instructions. Can you oh, you can't highlight it, huh? Okay, but oh, there you go. Perfect. Thank you. So it says right there, it says, read me that second line, Felipe.
All right. The second one, it
Yeah.
says the quiz non-graded.
Yeah.
Take the quiz non-graded.
Exactly. Does it say you have to take the quiz?
No.
No, it's up to you. I just put it there so that if you want to make sure that you understood the article, you can take it. But again, you have too many quizzes and too many assignments. So it's an optional thing. You don't have to do it. It also says highlight and annotate, not graded, meaning I know, Christian, you did the highlightings on some of them. So you don't have to highlight, okay? It's just there if it helps you. All I care about is that you do the writing prompt. Okay, so now go back, Christian, to back assignments. I'm sorry, go back to the article. I'm so sorry. Click on gene editing and then click on the article again. I don't want them to see your answers though. Click on activities. Click on activities. Okay, and then pause there. So I don't want you to click those, Christian, because then it's going to open up what you wrote and everyone's going to see your answers, and I don't want that. So from here, it gives you three options. It gives you ELA, which is English Language Arts. It gives you ELA Write, ELA Quiz, and then for science, it gives you a Write. You only have to do the ELA Write and the science Write. And by the way, Christian, I graded it and I sent you feedback on newsela.com. Did you get it? Don't click it right now because it's going to show all your answers to everyone. But do me a favor, Christian. Once this is done, read the feedback I wrote you. Okay.
Okay.
Okay. So you can exit your screen. Does that help, Felipe and everyone else? Give me thumbs up if that was helpful. Give me thumbs up if that was helpful. Good, Victor. Good, Felipe, Jessica, Daisy, Thanos, Denise, Vanessa, April. Good. All right, cool. Thank you, Dominic. Thank you, Naomi. All right, cool. So, and then Max. Good. So again, you can work on that at your own pace. So as soon as I'm done today, if we end early, you can go ahead and start it. If you're like, hey, Mr. T, I have some other work to do, I can't do the article, that's fine. Like I said, I'm not really worried about due dates. I just want to know, can you do these assignments on your own? So does anyone have any other questions that Christian or myself did not answer? Does anyone have any other questions about that reading assignment? I'll pause. Does anyone have any questions about that reading assignment?
No.
Okay, cool. All right, so we're going to go ahead and move on then, now that we know what the upcoming assignment is. And I'm going to go back to Schoology. So that's this, right? And then next Monday, and again, you can do them now, and a lot of you already did them. So for next Monday, I had told you guys that we're going to do a discussion board similar to the DNA, where you're going to write your own, and then everyone else's job is to try to solve them. And I had a lot of fun with that one, with the one that we did for DNA mutations. I read all your answers, and I posted comments for every single person. So please go back and read what I wrote you for the last discussion board on DNA mutations. Some of you tricked a lot of people. Some people weren't able to figure out all your point. I think it was, oh my gosh, who was it? I think Dominic and Denise had the most complicated. They put like 3 or 4 mutations in them, and a lot of people weren't able to figure all those out. So very good. I thought those were cool. So I did a similar one to this, where you're going to write your own DNA sequence. You're going to ask the class to try to solve the mRNA, the tRNA, and the amino acid. Okay. So here's the rubric as usual. And look, some people already did it. So thank you, Jessica. Thank you, April. Thank you. Okay, good. So you guys are the only ones that have done it so far. And again, it's not even due today, but I'm telling you and I'm showing you this so that you know you can work on these at your own pace. Okay. So those are posted. And then the quiz I had scheduled is for next Wednesday. If you feel ready for it as of today, let me know and I'll turn it on for you. Sound like a plan, guys?
Yep.
All right,
Yeah.
cool. All right, cool. So what we're going to do now is I want to wrap up this whole protein synthesis talk where we left off, which was the different types of mRNA. So in the video that we saw last time and in the watching assignment that you guys submitted to me, the question was, what's the difference between transcription and translation? And we heard some terms like mRNA and tRNA, okay? And I went over this with you on Wednesday, so if you weren't here, please watch that YouTube video. But essentially, there's 3 different kinds of RNA that are made for making proteins, okay? The mRNA, and first of all, where is mRNA made? Does anyone remember? Where do we make mRNA?
The nucleus?
In the nucleus, right? So the mRNA is made in the nucleus, and what it does is it copies DNA, but it rewrites it. And it rewrites it, and the reason we call it messenger is because it carries a message, and that message is called the codon. Codon. Think of code, okay? So it carries a special code. And that code, by the way, is a 3-base sequence, meaning it's made up of 3 letters. So example would be like AAA or AUG, okay? It's carried in a code, but it's only 3 letters long. That's what an mRNA is. The tRNA, which was mentioned in the video, is the thing, I think this was Max that told us last week, it's the thing that assembles the proteins, but what it does is it actually transfers or retrieves or picks up the amino acids. So does anyone remember where ribosomes are and where protein synthesis is made? Does anyone remember where that occurs?
In the
Say it, Vanessa?
in the cytosol?
In the cytoplasm or the it occurs in the cytosol or the cytoplasm, same thing. It occurs basically outside of the nucleus, right? That's where the ribosomes are, and that's where these amino acids are. And I think it was Max that told us, yes, it helps us to assemble the protein, but it has to go retrieve these amino acids throughout the cytoplasm, okay? And the way that it does that is it carries something on it called the anticodon. The anticodon essentially is also a 3-base sequence. Oops, sequence, but it's opposite of mRNA. Okay, so it's the exact opposite. So if mRNA is asking you for AUG, if mRNA is asking for AUG, what would the tRNA be, Max? If mRNA says, "Hey, we need AUG, Trujillo," what would the tRNA be, Max? If I'm saying I need mRNA codon AUG, what would the tRNA anticodon for that be?
I don't know.
Okay, so if I'm asking for AUG on the mRNA, what would so this is the mRNA, mRNA codon, okay? If I'm requesting that because DNA told me this is the code we need, then what would the tRNA anticodon be for that How about Noemi? What would the tRNA anticodon for AUG be?
UAC.
UAC. Is that correct, Max? Is that correct, Max?
Oh, my bad. How'd you get USC again?
That's a great question. Noemi, can you tell Max how you got UAC?
Yeah.
Well, A would be T, but then T turns to U. And then U is basically T, so U would turn to A, and then G would be C.
Okay, so
Okay.
very good, Noemi. So in case you forgot the rules, guys, I'll write it here, okay? But she's absolutely right. So the rules are that in DNA, you guys can see my pencil, right?
Yeah.
Okay, in DNA, like Noemi said, A's always pair up with what again, Denise? What does A always pair up with?
T.
T, right? And G always pairs up with what, Victor?
C.
With C. However, we're talking RNA now, right? This is RNA. So it's a different molecule. I went over on Wednesday that the molecule, its major difference is that it's a single strand rather than being a double strand. But Noemi brought up a really good point. A's normally pair up with T's, however, not in RNA. So instead of in an RNA, an A matching up with a T, Noemi told us that A's actually pair up with what again, Victor?
U.
Very good. And G still pairs up with C. You guys see that? Does that make sense,
Yeah.
guys? Okay.
Yeah, I
That's
got
how,
it now.
okay, good. So that's how Noemi was able to figure out that if I'm requesting the mRNA codon AUG, the tRNA anticodon has to be UAC, right?
Mm-hmm.
Okay, so let me get rid of my question mark so I don't confuse you guys, because I'm recording this and I don't want anyone to be confused. So very good. So I'm going to just erase these marks. And again, the video has all this stuff, so when you're done, if you need to rewatch anything, please do. But today I wanted us to get in groups and actually do some little problem solving, okay? So that's the mRNA codon. That's the tRNA anticodon. Now, in my notes, I go a little more in depth, but I basically just told you everything I need to tell you. That the mRNA, which is made in the nucleus, like April told us, it uses a new set of instructions like Noemi just summarized for us, right? It follows a new set of rules. In place of T's, there is a U, okay? And that code or message we call the codon, all right? And we just talked about that. The mRNA basically looks like this, okay? So DNA unravels itself like you saw in the video on Wednesday. And from this DNA template, we build a complementary new strand that we're going to call mRNA, okay? So here's what an mRNA looks like. The tRNA, like we just saw, is the opposite of what mRNA is requesting. So because it's the opposite, if I'm requesting the codon UUU, the anticodon is AAA. Does that make sense, guys?
Yes.
Okay, good. So that's the tRNA. The tRNA is this little, and we call it a tRNA, not just because it transfers, because it actually really looks like a T. It's this three-dimensional like strand that forms into like this coat hanger looking thing. And the reason that it does it is very important. You'll see that at the top, it has a place where the amino acids are going to attach to, right here. And you see that at the bottom, it has a place for the anticodon. So its structure is actually not like random. It's very, very important. So the amino acid will link up here and the anticodon is carried down here, okay? The ribosomal RNA or rRNA essentially it's just the ribosome. That's all it is. It's just a ribosome. We call it ribosomal RNA though, because what it is, it's made up of a bunch of bases. Don't worry about how many bases it is. It has like a little globular shape, which is what a ribosome is. And it is the actual place where protein synthesis occurs. So think of it like this. I gave you an analogy on Wednesday. I said, if we go back to this slide right here, DNA is the boss. DNA is the one that's requesting. Think of it like an engineer or an architect. DNA is saying, we need to build this house. Proteins are the house. So what would RNA be in that analogy? If DNA is the architect that says, we need to build this house, and proteins are the house that get built, what is RNA, guys? What's RNA in that analogy?
The builders.
They're the builders, guys. They're the guys that get all the stuff. They're the guys that read the instructions that the DNA sent. They're the ones that put everything together. That's essentially all that protein synthesis is. So again, these are the three different mRNAs, tRNAs, and rRNAs. Now, I just summarized for you what codons and anticodons are, so you don't have to read that slide. But now let's talk about codon wheels, okay? So we call the codon wheel the universal genetic code because it's the language that RNA uses. And get this, RNA and DNA is not a human thing. Every single living thing on this planet has DNA, from bullfrogs to blue whales to Trujillos to chinchillas, everything. And that universal code, which again is made from DNA, is universal. Everyone has it. But like we learned about in DNA mutations, sometimes the mutations can occur, right? If we inadvertently change a base, right? If we swap out an A for a T, well, that's a point mutation like we talked about, right? But if I delete an entire letter, what kind of mutation is that? If I delete an entire letter from a DNA sequence? What kind of mutation is that if I delete an entire letter from a sequence?
A shift mutation?
Something shift. Close. Something.
shift.
Thank you, Alexa. It's a frame shift. Very good, though, Vanessa. It's a frame shift. So the cool thing is our bodies are very smart. Our bodies avoid mutations, actually. Our bodies are smarter than we are because let's say, for example, all these mutations want to happen, right? Letters want to get swapped out. Letters want to get deleted. Well, and again, that can happen for various reasons. Mutations can be because of the environment, because of health, because of toxins, because of viruses like corona, right? There's lots of reasons why mutations can occur in our body. But our bodies are smart, and so our bodies avoid mutations. And the way it does it, we talked about briefly on Wednesday, and I want to talk about it more today. The codon wheel is the tool for us to look at that. So I sent you guys the codon wheel. Let me go back to your materials section. I sent you the codon wheel. You're going to use it today, okay? So I sent it to you under assignments, and you're going to open it up today. So you go to your module, you go to assignments, and that wheel is right here, okay? So here's your wheel. I sent you a video on how to use the wheel. It's pretty self-explanatory, and we've been using it, and Jessica and Alexa already finished their discussion board assignment, so clearly they know how to use it, and I'm assuming the rest of you guys probably have some questions in case you haven't done that assignment. So I sent you an instruction on how to do it, and we read it in class on Wednesday, so please watch that video on how to use it. So the codon wheel, like I just said, it makes it so that our bodies avoid mutations, okay? So let's say, for example, that I have a DNA sequence that reads, so here's my DNA, okay? Let's say my DNA reads A, U, A, T, I'll just do a short one, T, T, C, okay? That's my DNA. So what would the mRNA for this be? How about Daisy? For this DNA, what would the mRNA be for this? So here's my mRNA. What would the mRNA be?
UAU,
Okay,
Not
U, A, U,
AAG.
AAG, very good. Okay, so let's say, for example, let's see, who else do I have here? Let's say I swap out, let's say instead of making AUA, okay, so here's my mutated. Okay, so I'm going to mutate something here. Oh my gosh, sorry for my writing. Mutated DNA. Let's say I swap that though, okay? Instead of AUA, how about if my mRNA were to be U A hold on, where am I at?
Sound like you missed every shot.
What if I
You
swap
missed
it to
every
UAC? If my mRNA were to be UAC TTC. First of all, what kind of mutation just happened here where I swapped out this U for a C? What kind of mutation is that? It's a point mutation. Specifically, what kind of point mutation?
Substitution.
It's a substitution, right? I substituted just one base, okay? So the U turned into a C. Does that change the amino acid? How about Cynthia? Or actually, I don't think Cynthia has a mic. How about Daisy? If I swap out UAU to UAC, does that change the amino acid?
No.
Okay. So first of all, UAU codes for what amino acid?
Ty something.
UAU.
Tyrosine.
UAU, very good. Is we just say the first three letters, okay? So we can just say TYR, okay? But it's tyrosine. So the question was, if I swap out that U for a C, does it make a different amino acid? And you said no, right? Why do you say no? Why do you say no?
Because on the wheel, UAU and UNC code for the same thing.
Exactly. So check it out. UAC, it codes for the same exact thing. So going back to my slide, going back to this, can you guys see my wheel on my PowerPoint?
Yes.
Going back to this, the reason I say that it's redundant, do you guys know what redundant means?
Repetitive, kind of
Repetitive.
use.
Thank you, Vanessa. It means repetitive, okay? Repetitive. And the reason it's repetitive, like we just saw in that example, is to avoid mutations, right? Because if I swap out UAU and I change it to UAC, did that change the amino acid at all?
No.
No. So our DNA is smart and our bodies are smarter. We avoid mutations, we avoid errors because a lot of these, you can see, code for the same thing. There's only 20 amino acids on this wheel total, and believe it or not, every single living thing has the same exact amino acids. You, a bacteria, a blue whale, we all make our bodies from proteins using the exact same amino acids, everything. Okay, so the reason we need to know how to use this wheel, and like April and Jessica already did on the discussion board, is first, so you can understand how your bodies are made, like you saw in the PBS video from Wednesday, but two, so that you can understand what problems could occur as a result, okay? So what we're gonna do today is I'm gonna put you guys in Zoom groups, and like you saw in Wednesday's video, there are two parts to protein synthesis. We call it transcription, and we call it translation. Okay, so what I'm going to ask you guys to do in your Zoom groups today is I'm going to ask you in groups of about same, like we'll do 4 groups as usual. I want you to draw me what transcription and translation looks like in your own words. Okay, so you can look at the video again. You can look at my notes. My notes are actually very detailed and I include pictures of everything. So I'm going to let you decide how you want to do it. Okay, you can rewatch the video, you can use your book, you can use my notes, but I need, in your Zoom breakout groups, I need you to draw me or show me a model of what transcription and translation looks like. Clear?
Mm-hmm.
All right, thumbs up if you're ready. Thumbs up if you're ready. I'm waiting for a lot of people. You guys asleep or what?
No.
No. Waiting for Sydney, waiting for Jaime. All right, cool. All right, here we go, guys. So we're going in our groups now. And I want you to show me a model of what transcription and translation look like. So we'll do 4 groups. You're getting an invite from me now, and I'll see you in your groups. Bye, guys. See you in a bit.
No, I don't know if he wants like a actual example or like with the letters.
However you want, guys. Like I said, that's why you can use whatever source you want. You can do a drawing, you can use the letters, whatever works. I just want to see what transcription and translation look like. So I would actually use the letters, guys. That'd be a really good example so that I know you know what you're talking about. Okay?
Okay.
All right.
Yeah.
Thank you. Thank you.
How do we draw?
Yeah, I gotta figure out how we would do that.
I don't know.
Okay.
I'm gonna try. I'm gonna see if I can. It's called make a slideshow, and if you guys can write on it. Wait, is that still?
Maybe it'll work.
Yeah, if you guys have better ideas, just let me know.
Wait, Dante, how do you make it more gold? Is there another one today? Is there another concert soon? Is there another concert soon? Oh.
All right. Let me know if you can write on it.
Okay.
Yeah, you're not sharing your screen yet. So one of you needs to share your whiteboard with us. There we go. Perfect. So go ahead and write on it. Try writing on it.
Doesn't have the option for it.
Are
How do
you
you
on
do
a,
that?
Oh, you're on your phone, right?
Yes.
Okay. Is there anyone here that's on an iPad or a computer?
No.
No.
To a computer.
Okay. All of you are on phones?
Yeah.
Okay, go down to the bottom where it says share screen options and read me the options that pop up. Read me the options that pop up, guys. One of you.
There's
I think you could share a whiteboard.
Yeah. Is that the can you click on that?
Okay.
Okay. So go ahead and write on your screen, please, Daisy. You should have it. There you go. Okay, perfect.
Yeah.
So, okay, good. So what I need you guys to do is help Daisy come up with a model of transcription and translation.
All right.
Make sense?
Yeah.
Okay, thank you, guys. I'll be back.
I'm in erase mode now. Okay, there you go.
All right, cool. So who's drawing here? Noemi?
Yeah.
Okay, cool. So transcription, what does it look like? Good, good, good. So what are you drawing for us, Noemi?
Can you hear me?
Yeah.
I'm using your notes.
Okay.
So I'm gonna draw like the 2 strands.
Okay.
And the amino acids on all of them.
So what's this molecule you're drawing right here?
DNA.
DNA.
DNA, right? Okay, good.
Then
That's when they make DNA into RNA, right?
Oh yeah, I think so.
And so can you explain to us as you're drawing this, Nami, what are you illustrating here? So you put DNA strand, I'm assuming, right?
Yeah.
And then what are you going to do with these letters?
I'm gonna transcribe them, so I'm gonna do the other side too. And then there's like a parent strand, right? Like they like detach and then you make a new.
Yeah, so the parent or the original strand, Noemi, is going to be the part of the DNA you're going to use to make the mRNA. Does that make sense? So if you're using these letters here and you decide you're going to make a single strand from this, that would be called the parent or the template DNA. Does that make sense?
Yeah.
Um, Hi guys, so who's sharing the screen and who's drawing for us?
No one has shared their screen, and no one's really talking. But we're not really talking. We're kind of just chatting.
Okay, so that wasn't the assignment though. Someone needs to share their screen, please, because that's what's going to be on the quiz is transcription and translation. So the reason I'm giving you this opportunity to draw it for me is so that I can help address any like misconceptions you have. So if you guys don't draw it correctly, I can tell you what you did wrong. But if you don't draw it at all and then you get to the quiz, you're not going to know what you're talking about. So who's going to share the screen, please?
How do we share the screen? Because last time it didn't let me.
You go to your share screen options, April. Click on the share options.
I don't share content.
Yeah, click on that. What does it tell you?
It says screen, photos, iCloud Drive. Do I click on screen?
What other options does it have? Does it have whiteboard?
Yeah.
Okay, click on that. Click on whiteboard. There it is. Okay, go ahead and just do a little squiggly line, April, so you know that it works.
Oh, wait. Let me see. Oh, okay.
There you go. Okay, so what I need you guys to do, and I'm going to be back, and if this is not done, that means you guys are not listening to what I'm asking you to do. So Christian, Sydney, and Thanos. I know Thanos and Sydney don't usually have mics, but they can still chat with you, and they can actually write on your screen. Do me a favor, Sydney, can you write on April's screen right now, please? You have a little pencil feature. You can all write on this screen.
It doesn't give me the option to.
It doesn't give you the option to? What do you have on your screen right now, Sydney?
It's just the whiteboard.
And you can't draw on it?
No.
You don't have like a little pencil or an annotate feature? Christian, do you have an annotate or a pencil feature?
No, it's just like the regular thing.
Okay. Who just drew that line right now?
Oh, that was me.
It was you? Okay. All right, then tell you what. April, you draw it, but I want you to draw it with the collaboration of Sydney, Thanos, and Christian. Okay? So transcription. You already told us, April, that this happens in the nucleus. So how can you show us transcription? What is transcription in your own words, guys? Because all of you sent me your answers on the watching assignment. So what is transcription?
Isn't that where the RNA what is it? It takes away like the wait.
Isn't it when it makes tRNA?
Okay, so then what would that look like?
I think like isn't the DNA supposed to be like split up?
Yep, it is. So that would be a good place to start, April. So you guys give her instructions and she'll draw it. Okay? Okay? Hello?
And then I think the RNA is supposed to go in and it like sorts itself into the DNA.
So what would that look like?
Wouldn't this be like a DNA but like split?
Okay, so what would that look like? Yeah, so what would that look like?
Like, wait, this looks better now. Just go with the process. Wait. And then, and then
Okay. So can you? Okay, good. So can you pause right there, April?
Mhm.
Okay. So let me erase something for you. Okay. You don't need to show us the directions right now. Can you erase that arrow that you just put?
There.
Okay. So check this out. First of all, what is this thing that April drew us unwinding or opening up? What is this molecule? What is this thing that April just drew us? What is this thing?
Strand?
Huh?
A DNA strand.
Very good. It's just DNA. You can see my writing, right, guys?
Yes.
Who's writing right now? Who just wrote that?
Wrote what? The DNA?
Yeah.
Oh, I don't know. I didn't.
Okay. So this right here, guys, this is the DNA strand, okay? And so that DNA strand, what April drew for you is it opened up, okay? And so let's say, for example, that the DNA has the following letters. Can you guys see my letters?
Mm-hmm.
Are they on the DNA or are they somewhere else?
They're somewhere else.
Oh, okay, yeah, it's somewhere else for me too. I don't know why it's showing it up different. Tell you what, April, can you erase this whole thing? Because for some reason, what I writes it in a different place as you. Can you erase the whole page? And I'm going to show you something, okay?
Erase everything?
Everything, yeah.
Okay.
Okay, just, yeah, everything. Okay, cool. So I'm going to redraw what you drew, April, okay? So here is your DNA. And I'm going to draw exactly what you guys just told me. What Christian said is that there's a DNA, and remember it has the base pairs, but the base pairs are going to unzip. They're going to open up. Someone has a loud bird. Whose bird is that?
I have 2 birds. I'm sorry.
Oh my gosh. Okay. So there's my DNA molecule, and there's the DNA opening up. You guys see that, right?
Yeah.
So for some reason, it's off because when I drew it, it looks different than what you guys see. But when it opens up, remember there's letters in here. There's little letters in here, and those are the bases. So if we have an A, a T, and a C, and I want to make a DNA, an mRNA from that. It looks so weird on my screen. When I draw it, I think it looks different for you guys. But let's say that I have an A, a T, and a C on my DNA. Okay, this is my DNA. Right, and all this is represented on the model that April and I drew. So if that's the DNA and we're going to make an mRNA from that, all we're going to do is we're going to match up using the new rules. Remember, Noemi told us A's and T's don't exist in the world of RNA. It's A's and U's matching up. So if that's our DNA, what would the mRNA for this be?
UAG?
Okay, very good. And you would just keep doing that as long as the DNA is requesting for the code. So if the code is like 10 bases or 1,000 bases, you keep doing the little letter coding as long as you need to. That little code, that code is called the codon. That new rewritten message that mRNA writes from the DNA is called the codon. Does that make sense?
Yes.
That's all that transcription is. So I'm going to erase all this now. And I'm going to ask that you guys show me your own version of what that looks like. You can use the letters. You don't have to draw a DNA unsplitting. I know that Christian started talking about it and you drew it, April, too, but you don't have to. You can do whatever it is you want to do. Okay? Sound good?
Yeah.
Okay, I'll be back. All right,
Hi.
last group. How's it going here?
Oh, good. We kind of like explained it,
Okay.
but we don't know how to like draw on it because it doesn't allow us.
I tried sharing my screen, but it didn't work.
Really? Okay,
Yeah.
tell you what. Let me share my screen. Are you on a computer, iPad, or phone, Melissa?
I'm on my phone.
Okay, that's what I think the problem is. It works on the phone because I just did it with April's group, Melissa, and it works. It's kind of funky on a phone. So I'll share my screen, and why don't you guys tell me what to draw? Okay. So here's my screen. What do I draw?
Oh wait, we can draw now.
You can?
If we can only draw when you share your screen.
Oh, that's weird. Okay, so then draw on here and show me what you guys were talking about since I haven't seen your group.
So, okay. How can I draw? Ugh. Just gonna draw one big circle.
Okay. And what's this big circle representing?
The entire cell.
Oh,
The cell?
God.
The cell? Okay. So what happens here?
And then we have, hold on.
What about the cytoplasm?
And then the nucleus.
Okay. So we have the locations, but what actually happens here?
Oh, okay. So inside the nucleus is where like the DNA is held, and so for like to make RNA, they have like this protein making machine inside the nucleus. So it starts out here. Oh dear. And then so it just kind of like splits apart the bonds of the DNA that kind of like holds it together.
Okay, and what does that look like?
It's like a
Draw it for me.
Oh dear. My drawing's terrible on the phone.
Or
Oh, okay.
Someone can draw.
Yeah.
Okay, Vivian, that's fine.
Okay.
Who can help out Denise here? So we have the nucleus, we have the cell, the cytoplasm is all the blank stuff, right? So let's zoom in only to the nucleus, Denise, because that's where transcription occurs. So let's just look at the green thing. So show us, you just said that the DNA opens up. So what does that look like? So you can erase all this. We just want to see the nucleus, what's inside the nucleus. So what you said that the DNA opens up. So what does that look like?
Like it's in like this kind of machine that the DNA goes through. God, I can't draw. But then it kind of like starts separating them, like.
Okay. Okay, so it opens up the DNA.
And then like some of the RNA bonds, like the RNA, what's it called? Building blocks. Like start coming in. Hold on, let me use a different color.
Okay.
It's like these little things start like swarming in, and then they attach in one of the parental stands. Like each one to the corresponding one.
Mm-hmm. Yeah, those are called the primers. We talked about those before. Okay.
Yeah.
And then what happens? So let's say, for example, I'm going to pick it up where you started, Denise. This is a good starting point. So you said this little machine, and this is called, I haven't talked about that enzyme. It's called RNA polymerase, but for now, we'll just call it that machine. You're right. It opens up the DNA like you unzipped it here. And I'm just going to zoom into your unzipping. Okay, so your unzipping part, which is right here. right? You said it opens up. Okay, very good. And let's say it has, let's put some letters so that we can make this a little more, you know, like related to the wheel. Let's say, for example, we have these letters right here. Okay, we have A, we have a T, we have another A. Oh, that's an A. And then we have a C. Okay. And then over here, so this is one part of my DNA, this is the other part of the DNA. So if this part of the DNA read ATAC, what would this part of the DNA be, Jessica?
It'd be for the mRNA.
No, we're still doing, we're just looking at the other side of the DNA. We're not doing mRNA.
Oh, okay. Okay. It'd be GTAT.
Yeah, G, T, A, and T. Very good. So that's the DNA. And I'll do this in a different color so that you know the difference. So this here is just DNA. Okay, so here's the DNA. And then here is another DNA. And all we did, like Denise told us, is we just split it up with this little circle thing, okay? But now we want to do what Jessica just said. We now want to make what's called the mRNA. So let's say we use the bottom half, okay, the GTAT. And now I want to make my mRNA. So we need to know now for this new molecule, which we're going to call mRNA, we want to know, well, what letters would match up to this? So can you tell us, please? Go ahead.
CAUA.
CAUA. Do you guys agree with that?
Yep.
Yep, that is absolutely right. But how many letters did I say make up one codon?
3.
3. So all of this would not be used, actually. We'd only use 3 letters at a time. So let's say, for example, we're only going to use this, okay? This little short segment of 3 is what we call the codon. And all of this, guys, what you just drew right here with me, this is called transcription. This is transcription. Oh my gosh. Transcription. And can you summarize for us, please? How about Melissa? What is it that we drew for transcription here? What is transcription?
Isn't it when the DNA gets copied into mRNA?
Exactly. And is that not what we just drew?
Yeah.
It is, right? Exactly. It's simple as that. So I know that video made it very complicated. It's simple as this, guys. The DNA opens up like Denise drew, and then all I did was finish up Denise's drawing. I just put some letters in there, and Jessica and Melissa just helped me figure out the mRNA. Thumbs up?
Thumbs up.
All right, cool. So now, we're not done though. So now, what is translation? So I'm going to hand you over the pencil. Who can draw us translation? I'm just going to put the title. Who can draw us translation? So first things first, what do we need to do translation?
The mRNA.
The mRNA, right? So I'm going to rewrite my mRNA for you. I'll get you started. And my mRNA, we already wrote it here, right? We had said it was AUA, but that's too easy, right? Let's make a longer one than that. It's kind of boring. So we'll just make up some more letters. So AUA, give me some more codons, guys, and sequences of 3. So AUA, make up some more.
UCG.
Okay, UCG. And how about give me one more?
Oh, okay.
AU.
Yeah.
AU what?
A.
AUA. Okay, so do me a favor. If that's the mRNA, what do we need now?
tRNA.
The tRNA. Oh, yeah, you're absolutely right. I'll do it in a different color. So what would the tRNA for what you guys just wrote, what would the matching tRNA for that be?
WAU.
UAU. Okay.
AGC.
AGC.
And a UAU.
And a UAU. Do we agree with that?
Yep.
Okay. Are we done?
No.
No. What do we need?
The amino acids.
Amino acids, very good. So now we need to figure out amino acids. So this is where you guys come in. Everyone, please open up the codon wheel that I sent you on Schoology and tell me the 3 amino acids for this. This is your job now. What are the 3 amino acids here? I'll leave this drawing up, but you guys find me the amino acids.
Okay. U A U. Okay, U A U. U A U. T Y R.
Okay. So we would write T Y R. We just put the first 3 letters. And then we put a little line to show that they're connecting, because remember, we're making a chain of amino acids, and one amino acid connects to the other till we get that 3-dimensional protein. So what goes next? How about Melissa? So TYR is the first one. So what's AGC, Melissa? Are you there, Melissa? Or are you?
I can't find it.
You can't find it? Okay. Can someone help out? What's AGC?
It'd be SER.
Yeah.
What is it?
SER.
SER. Okay, very good. SER. And what would the last one be?
TYR.
TYR. It repeats. And that's the point of this. Remember, the code, whatever is being requested from the RNA, that came from the DNA. So the DNA confirms, it repeats letters like you just saw, AUA, AUA. So it's natural for the amino acids to repeat. So this sequence of amino acids, this right here, this is the protein that we just built, okay? And that's all that translation is. Does that make sense, guys?
Yep.
Someone's got an angry dog. Whose angry dog is that? Okay, so I'm going to go ahead and save this picture for us so that I can share it with everyone else. But good job, guys. Oh, first of all, translation, where does this take place? Where does all of this stuff that you just wrote me take place?
In a
The
ribosome?
cytoplasm.
In the cytoplasm, but specifically where?
In a ribosome?
Very good. So all this thing that I'm drawing, this little brown structure, is called the ribo. Absolutely. Good job, guys. So I'm going to take a picture of this thing and save it for all of us to see. And I'm going to go ahead and break up the groups now, okay? Good job, guys.
Okay. Thank you.
All right, guys, welcome back. Welcome back. So I went to every single group, and some of them took a little longer than others, but we were trying to draw what transcription and translation looks like. So if you didn't get a chance to finish it, I was just in Melissa's group, and we drew the whole thing out. So please make sure you watch the YouTube video that I'm going to post at the end of this class so that you know what you missed. But from the other groups, I know the first group I was in, I believe was, I think it was Noemi's group. Were you guys able to do your drawing, Noemi?
Yeah.
Okay. Did you take a screenshot of it, or did you erase it?
I didn't.
You didn't? Okay. Well, tell you what. It's okay because I have it here. We'll tell you what I'll do is at the end of this, in the YouTube video, I'm gonna post a picture of what Denise and I was in Denise's group, and she started doing the drawing, and then I helped to finish it. So I'll put up a picture of that drawing so you guys can see what it is. But I'm gonna go ahead and wrap up here if you have no other questions. But I went to all the groups, and I think for the most part, we all understood kind of what this looks like. So let me just ask before I end class. In your own words, Noemi's group, what's the major difference between transcription and translation? Anyone in Noemi's group?
Well, in translation, I guess it's more based on the amino acids
Okay. And what do we use those amino acids for?
to make the protein.
To make the protein, right? And what happens in transcription, in your own words?
Makes RNA.
It makes RNA, but which RNA, Jaime?
T RNA
And
mRNA.
And the mRNA, very good. And what's the major difference between mRNA and tRNA, Jaime? What do they use for? And then we'll pause right there.
Needs a little help.
Yeah, anyone can help out.
Beeps, you wanna help me, buddy?
Help them out.
I know how mRNA and tRNA is used during translation.
Okay.
So in the ribosome, they're both used to they're both used to make a sequence of amino acid.
Mm-hmm.
According to I think it was tRNA's sequence, like its coding.
Okay. Okay. Yeah, basically, and I'm going to just show you a picture, and I know I'm over by a few minutes, so I'm just going to show you a picture to wrap all this up because we pretty much covered the whole thing. So let me just share with you one last picture, and then I'll call it a day, okay? So here's what transcription is, right? So this is pretty much what Jaime and Noemi were telling us, okay? So transcription, and I went to all your groups, and you guys showed it to me differently. Some of you did letters. Some groups drew a DNA unwinding. So whatever you drew, this is what happens, okay? We have the DNA opening up, okay? So you can see the blue is the DNA. It opens up, and then what's being made, which is a single strand, is this little pink RNA, okay? So if the DNA reads GTA, GGTTA, the matching mRNA is what then? Someone read it.
CAUCCAAU.
Very good. So very good. That's all that mRNA is, and that's all that transcription is, okay? Translation, on the other hand, let me just go real quick. This is translation, okay? It happens like this. This is the mRNA, okay? The mRNA is this AUG, CUA, CUU, CGA. And remember, they're increments of 3. That's called the codon. The mRNA enters into this big little chubby thing that we call the ribosome. This is the ribosome, okay? So the mRNA brings the code. The tRNA brings the what again? What does the tRNA bring us?
The amino acids.
The amino acid and the anticodon, Vanessa. So yes, it has the amino acid at the top. The anticodon is the opposite, like I told you, to the codon. So if the codon is AUG, the anticodon is what? If the codon is AUG, what's the anticodon to that?
UAC.
UAC, like you see here, right? So this UAC brings us this amino acid, okay? Then the next codon is CUA, okay? And that CUA, if you have your codon wheels, you can figure out what that amino acid would be. So amino acid 1 links to another amino acid. What would the next codon on this mRNA be, please? How about Jessica? What would the next codon be?
Which one?
Well, okay, so I read the first 2 codons. It was AUG,
Yeah.
CUA. What would the next codon be?
CUU.
Very good. It'd be C, U, and U. Because how many letters are in a codon, Jessica?
3.
There's only 3. So it'd be the next 3. So then CUU would bring its next codon, and you just keep doing this over and over and over and over and over until we get to what's called the stop codon. The stop, let me just show you this, and I'm done right here, guys. The stop codon, if you look at your codon wheel, there's only 3 amino acids that tell the ribosome, hey, you can stop. We've made the protein we need to make. So when the codon reaches the end of the line or it reaches the stop codon, it's done. Just like this class, we're finished. So on this codon wheel, what are the stop codons, guys? Can anyone find them for me? And I'm going to end right here. What are the stop codons? Who can find them on the wheel?
UGA.
Okay, U, G, A. What else?
UAA.
Very good. U, A, A. And?
UAG.
Very good. U, A, G. So on that example I just showed you, if it reaches any of those, is there a stop codon on here? And if so, what is it? Is there a stop codon on here? And what is it?
Yes, it's UAG.
It's UAG, right? It's right here, guys. Look, UAG. Cool? You with me, guys? Does this make sense?
Yes.
All right,
Yeah.
cool. So sorry, guys, class went a little late. I'll go ahead and post this YouTube video if you want to rewatch anything, but we're officially done. So if you haven't already done so, I'm giving you the day to do the reading assignment. Thank you, Christian, for showing us how to do it. Again, I know you have other homework, other lives. Don't stress if you can't do it today. Just message me. Let me know when you do it. I get them directly, so don't worry. But please, when you do them, the next assignment is the discussion board that Jessica and April already did. All right, so if you have no other questions, we'll call it a day, and I'll see you on Monday. Cool?
Yeah.
All right.
See ya.
Bye,
All right,
guys.
later, Mr.
Have a nice weekend, y'all.
Bye.
Bye.